Welcome to the review on genetic technology. This is going to go over artificial selection and genetic manipulation, meaning people choosing what traits get passed on and or people mixing and matching DNA. For that, we're going to take a look at cloning, selective breeding, genetic engineering, also called gene splicing or bioengineering, and gel electrophoresis. The first of these we're going to look at is cloning. And cloning is when you make an identical copy of an organism. There are some pluses and minuses we'll go over, and it's a very straightforward process. Looking at our little setup, we have elephant A, B, and C. A and B are necessary. C, you might need it, you might not. And let's take a look why. Elephant A, we're going to grab a cell from this elephant. This cell has to be a diploid cell. Body cells are diploid, therefore it's going to be a diploid body cell. Diploid means di, two. That refers to having two chromosomes for each gene. One from mom, one from dad. Right here, we have two chromosomes on here. This is from mom, this one's from dad. That makes up your body cells or this elephant's body cells. From elephant B, we're going to need a different cell. We're going to need a monoploid egg cell from B. Mono means one. Therefore, they don't have two chromosomes for each gene. They have one chromosome. And this chromosome combines with the one chromosome with genes A and R from the male to make a new diploid cell, a new baby in sexual reproduction. B, therefore, since we're getting an egg cell from it, it must be a female. Egg cell is also called an ovum. Elephant A could be male or female. Doesn't matter. We're making a clone of it. We have all its DNA. Doesn't matter. We're going to use the empty egg cell from B, and we're going to use the diploid nucleus, the full complement of chromosomes from A. That means that empty diploid cell is going to go in the garbage. That monoploid nucleus from the egg cell, garbage. So now we're left with a zygote. Well, a sperm didn't hit it. It didn't get fertilized. No, we artificially did this. Whenever an egg cell becomes diploid, meaning when a sperm hits it and it goes from monoploid to diploid, it becomes a zygote. Well, we just did something a little different. We just took a diploid nucleus and shoved it into an empty egg cell. Therefore, all the DNA came from A. This nucleus now, this cell, this zygote, is going to be put into elephant C. Elephant C will allow this growing ball of cells, which will become an embryo and a fetus and eventually a baby elephant. It will allow it to grow inside its uterus. Therefore, elephant C has to be a female because it needs a uterus, and elephant C is called a surrogate. It will grow a foreign baby inside her uterus. And 18 months later, for elephants, you get a new baby. This baby contains all the DNA from elephant A. Therefore, the baby that's born is a clone or identical copy of elephant A. And the reason, we kind of said this a little bit, all the DNA that's used to make the clone, it came from A. It came from A's diploid cell, which is the full amount of DNA shown in the pictures above of the chromosomes. Question. If you want to clone a specific animal, what do you need? Well, if you want to make a clone of yourself, you need a body cell from yourself or a sex cell from yourself. You need a body cell because that has all the DNA to make you. What if you tried to make a clone of yourself with a sperm cell or an egg cell? Well, you're only going to half of your DNA. Now, you don't want to make half of yourself. It's impossible. If you try to fertilize an egg, you can't make a copy of yourself using half the amount of DNA. 23 chromosomes won't grow into a baby. If that's the case, then an egg cell will magically start developing into a baby without the need for sperm. Therefore, if you try to take a sperm cell and make a clone of yourself, you have to combine that monoploid nucleus with only half the amount of chromosomes. You got to combine it with the sperm cell, or vice versa. If you have a sperm cell with only half your number of chromosomes, you're going to have to combine it with another half from an egg cell to make a diploid cell. Therefore, that won't be a clone of you. That's why you need a diploid nucleus from your body. And there's our explanation. If you try to use a monoploid cell from the individual you want to clone, you'll only have half the amount of chromosomes or DNA, and you would need to combine it with another individual's monoploid DNA. Not going to make a clone. You also need an egg cell to put the diploid nucleus, and you need possibly a surrogate. Possibly because if a female is cloning herself, she could technically put her clone, that little fertilized, or that little zygote, I keep saying fertilized egg, the zygote, that's a clone of her. She could put it inside herself, in her uterus, have that baby grow, and 9 months later, she can give birth to herself. A little bizarre. Some positives and negatives. If you have a couple of rhinos left, like 2 or 3, and they're on the verge of extinction, you can clone these things and increase the numbers that help revive the population. Granted, there's some drawbacks, but nonetheless, it's better than going extinct. So, save species from extinction. 2, you can make more organisms, identical copies, that have the desired traits that you want. So if you finally get, let's say, a strawberry plant that is growing really big strawberries and they're super sweet, there must have been some cross-pollination event or something that went on to give you this plant. But you don't want this plant to die at the end of the season, so you clone that plant. You could clone that plant and make all your strawberry plants big and sweet. Same thing if you have a prize-winning racehorse, really fast horse that got born. You want to take some cells and clone that thing so you can have generations of identical horses and constantly have a very fast horse. Some negatives. The Irish found out the negatives the hard way. They took a potato plant, cut it up into pieces, planted all those pieces, got all identical plants. They kept doing this time and time again, and all the potato plants were identical. A fungus came along, killed one plant. If one was susceptible or able to die, all of them died. That led to the Irish potato famine, all because there was no variation. And that could lead to the extinction or decimation of an entire population of species, just because there's no variation. That's cloning. Stay tuned for the next video.